My name is Rich Valentis. I am the general manager here at Catachem. Joined the company just over 2 years ago. My background is mainly in molecular biology. I've spent the last 15 years of my career in CLIA, JCO-regulated laboratories. Did some biotech before joining the medical device industry. And yeah, happy to be here and thank you for joining. Some quick introductions about Catachem, and then we'll start the pre-recorded webinar. The voice you'll be hearing on the webinar is David Templeton. He's our CSO. He is on PTO. He's down in sunny Florida this week. So you guys are stuck with me, unfortunately. Catachem has been in business since early 80s, 1981 to 1984. I'm not sure of the exact date, but somewhere in there. They started as a medical device manufacturer, or we specialize in liquid chemistries, your general AST, ALT, liver enzymes, glucose, general chemistries for manual and automated assays. A large part of our market share is in the veterinary industry. We also have a select few products that we are putting through the FDA for human use, and we have several products in human use for RUO only currently. Our total catalog is right around 80 products. You can take a look at our website if you're interested in any of those. We've been performing lyophilization internally for the past 15 years on some of our calibrators and controls. And just recently we started, and by recently, the last 4 or 5 years, taking on outside contracts for lyophilization and kind of a CDMO / contract manufacturing style. Yeah, I think that's about it. We'll have a Q&A session at the end, and I hope you guys enjoy the webinar we put together for you.
Hello, everyone, and thank you for joining today's webinar. From Development to Defensibility, How Catachem De-Risks Lyophilization. My name is David Templeton, and I'm the Chief Scientific Officer at Catachem. Today we'll discuss lyophilization not just from a scientific standpoint, but from a development and risk management perspective. Many scientists understand the technical fundamentals of freeze drying, but what organizations often struggle with is translating those fundamentals into a process that is scalable, documented, reproducible, and defensible. In biotechnology, diagnostics, medical devices, and pharmaceutical development, technical success alone is not enough. A formulation that works once in the lab still carries significant risk until it becomes a controlled manufacturing process. Today we'll walk through both the science of lyophilization and how Catachem approaches risk reduction under an ISO 13485 quality system. Let's start with an overview of what we'll cover today. First, we'll discuss lyophilization itself, what it is, how it works, and why it remains the gold standard for stabilizing sensitive biological materials. Next, we'll review the key stages of the process. After that, we'll introduce risk, where it enters lyophilization development and how it can affect scale-up and product performance. We'll then discuss risk assessment tools and how organizations evaluate potential failure points. Following that, we'll look at risk management strategies and how ISO 13485 quality systems support controlled development. Finally, we'll discuss documentation, post-market monitoring, and how these practices help ensure defensibility. Before we discuss the freeze-drying process itself, it's useful to define what we mean by risk in life science development. Across biotechnology, pharmaceutical, and diagnostic industries, risk typically falls into 3 broad categories: technical risk, manufacturing risk, and regulatory risk. Technical risk asks a simple question: will the product perform consistently when the end user reconstitutes it? For example, a protein standard might show excellent activity immediately after lyophilization, but lose potency after 6 months because the residual moisture was slightly too high. A diagnostic calibrator might reconstitute differently depending on cake morphology, leading to inconsistent instrument readings. Or a nucleic acid reagent might degrade if oxygen exposure during stoppering wasn't properly controlled. These issues are often subtle, and they may not appear until stability studies or field use reveal them. The second category is manufacturing risk. This addresses whether the process that works in the lab will still work when scaled to production. A cycle developed for a few dozen vials may behave very differently when thousands of vials are loaded into a larger freeze dryer. Thermal mass increases, heat transfer changes, and product temperature may vary across shelves. Fill depth, vial geometry, and loading patterns can all influence sublimation rates and residual moisture distribution. Without careful scale-up work, a process that works beautifully in R&D can become unstable during commercial production. The third category is regulatory risk. In regulated environments, success is not only defined by the product working, it must also be defensible. This means the process must be documented, traceable, and reproducible. Auditors and regulators may ask questions such as, how were the product's thermal limits determined? How is residual moisture controlled and verified? What documentation demonstrates that each lot was manufactured consistently? If those answers are not supported by controlled records and validated processes, the product may face regulatory challenges even if it performs well technically. Development work is therefore about converting unknown variables into controlled variables. The goal is not simply to make a product work once. The goal is to build a process that consistently produces the same result, scientifically, operationally, as well as from a regulatory standpoint. Lyophilization remains the gold standard for stabilizing many biological materials. The primary reason is that it removes water gently. By freezing the product and removing water through sublimation, the process avoids the high temperatures and mechanical stresses associated with other drying methods. This helps preserve the activity of proteins, enzymes, antibodies, nucleic acids, and diagnostic reagents. Equally important, lyophilization is a predictable and documentable process, which makes it well suited for regulated environments. Lyophilization can be summarized as a sequence of controlled steps. First, the product is frozen. Then, primary drying removes ice through sublimation. Secondary drying removes bound water remaining in the matrix. Finally, the product is sealed and packaged for storage and transport. While this description is simple, the engineering required to control these steps is complex. Small variations in temperature, pressure, or formulation can significantly influence the final product. The first stage of lyophilization is freezing. Freezing determines the structure of the ice crystals, and that structure becomes the pore network left behind after sublimation. Large ice crystals create larger pores and lower resistance to vapor flow. Freezing too quickly can produce very small pores and slow the drying process. Freezing too slowly can cause phase separation in certain formulations. Parameters such as freezing rate, nucleation behavior, and annealing can all influence the final product structure. At Catachem, we treat freezing as a design step rather than simply a preparatory step. Primary drying removes the majority of the water. During this stage, ice sublimates under vacuum while heat is supplied through the lyophilizer shelves. The most important variable here is product temperature. The product must remain below its collapse temperature for amorphous systems, or below the eutectic melting point for crystalline systems. If the product exceeds these limits, the dried structure can collapse. Our goal is to operate at the highest temperature that remains safely below these thermal limits. Collapse temperature and eutectic temperature define the safe operating limits of the process. The collapse temperature represents the point where an amorphous formulation loses structural integrity. The eutectic temperature represents the melting point of crystalline components. Exceeding these limits can lead to structural collapse, inconsistent moisture levels, and reduced long-term stability. Collapse is not simply cosmetic. It can signal deeper structural damage in the dried matrix. Secondary drying removes bound water remaining in the product matrix. This stage often determines the final shelf life of the product. Shelf temperature is increased under vacuum to drive desorption of residual moisture. However, temperatures must remain within limits for the active ingredient and the excipient system. At Catachem, we define moisture endpoints through analytical measurement rather than relying only on process time. Once drying is complete, the product must be protected from environmental exposure. Lyophilized products are extremely hygroscopic and can absorb moisture if exposed to air. Vacuum stoppering can minimize oxygen exposure. Nitrogen backfilling replaces oxygen with an inert atmosphere. Container closure integrity becomes critical because even small leaks can allow moisture or oxygen to enter over time. Residual moisture is one of the most important quality attributes in a freeze-dried product. Too much moisture accelerates degradation reactions. Too little moisture can destabilize certain proteins. The objective is not to achieve the lowest possible moisture level, but to define a validated moisture range that preserves product stability. Uniformity across the load is also essential. Edge vials and center vials must produce consistent results. Even though lyophilization is well understood scientifically, risk can still enter development in several ways. One of the most common sources of risk is incomplete characterization of the formulation itself. If thermal limits such as collapse temperature or eutectic temperature are not fully understood, the process may operate too close to those limits, leading to structural collapse or inconsistent product performance. Scale-up is another major risk point. A cycle that works well for a small number of vials in R&D may behave quite differently when thousands of vials are loaded into a production-scale freeze dryer. Changes in thermal mass, heat transfer, and vapor flow resistance can create temperature gradients that affect drying uniformity. Moisture endpoints are also critical. If the acceptable residual moisture range is not clearly defined and validated against stability data, the product may appear dry but still degrade over time. Container closure compatibility can introduce additional risk. Stoppers, vial geometry, and sealing methods must be compatible with the process and with long-term storage conditions. Even small leaks or oxygen ingress can compromise stability months after manufacturing. Another common risk factor is insufficient process monitoring. If product temperature, pressure stability, or moisture trends are not tracked carefully during development, subtle problems can go unnoticed until later stages. Finally, documentation itself can become a risk point. If development work is not properly recorded, it becomes difficult to demonstrate how the process was designed, optimized, and controlled. In regulated environments, undocumented success is still risk. A process must not only work, it must be explainable, reproducible, and supported by controlled records. Organizations commonly use structured methods to evaluate risk. Failure Mode and Effects Analysis, or FMEA, identifies possible failure points and ranks them based on probability and severity. Risk matrices help prioritize risks based on impact. Hazard analysis frameworks are also used, particularly in medical device development. These tools convert unknown risks into documented engineering decisions. Once risks are identified, mitigation strategies can be implemented. In lyophilization development, this often includes thermal characterization of the formulation, defining moisture endpoints tied to stability studies, mapping shelf temperature distributions, validating probe placement, and maintaining controlled batch records. The objective is to reduce variability and demonstrate reproducibility. At Catachem, we operate under an ISO 13485 quality management system. ISO 13485 provides a structured framework for developing and manufacturing products intended for regulated life science and diagnostic environments. It ensures that development work is performed in a controlled and traceable manner rather than through informal experimentation. This framework supports disciplined development through controlled procedures, full traceability of raw materials and intermediates, formal design and development controls, and change management processes. It also encompasses documented training and competency requirements for personnel. Vendor qualification and raw material approval processes ensure that critical inputs are sourced from evaluated suppliers and meet defined specifications before being used in development or manufacturing. It also ensures that critical development decisions such as formulation choices, process parameters, and acceptance criteria are documented and maintained within the quality system. The result is that development knowledge does not remain with individual scientists or engineers. Instead, it becomes institutional knowledge embedded in controlled documentation and standard operating procedures. This allows processes to be reproduced consistently, transferred between teams or facilities, and defended during audits or regulatory review. Documentation is often overlooked as a risk management tool, but it is essential in regulated environments. Device master records, batch records, validation reports, raw material traceability, and stability data all contribute to a defensible manufacturing process. These records also support product conformance by clearly defining the acceptance criteria used to evaluate each batch, often developed collaboratively with customers to ensure they reflect the intended application and performance expectations. If a product must later be reviewed by auditors or regulators, this documentation provides the evidence that the system is controlled. Risk management does not end when a product is released. Post-market monitoring helps ensure that products continue performing as expected under real-world conditions. This monitoring includes structured review of customer feedback, quality trend analysis of production, and QC data, CAPA investigations when deviations occur, and field corrective actions when necessary. These activities help identify emerging patterns that may not be visible during development or validation. For example, a manufacturer might observe through QC trending that residual moisture values are slowly drifting upward over several production lots, even though they remain within specification. That trend could indicate subtle changes in loading patterns, stopper performance, or environmental conditions. By detecting the trend early through post-market monitoring, the manufacturer can investigate the root cause and correct the process before product performance is affected. These systems allow potential issues to be identified early, addressed systematically, and documented within the quality management system. Lyophilized products can be produced in several formats. Traditional vial formats are common for diagnostics and reagents. Bulk trays may be used for intermediate materials before final packaging. Bead formats are useful for automated workflows and high throughput assays. Selecting the correct format early in development can prevent costly redesign later. Making that decision early also helps mitigate scale-up and performance risk by ensuring the formulation, drying cycle, and end-user workflow are aligned from the beginning. During lyophilization development, several variables are monitored closely to reduce technical and manufacturing risk. These include product temperature, chamber pressure, residual moisture, cake morphology, and reconstitution performance. Tracking these parameters helps confirm the product remains within defined limits and allows potential issues such as collapse, uneven drying, or instability to be identified early. Together, these variables provide measurable indicators of product stability, process reliability, and overall risk mitigation. Our development workflow follows a structured sequence designed to identify, evaluate, and mitigate risk throughout the lyophilization process. First, we characterize the formulation's critical thermal limits to establish safe operating boundaries. Next, the process is developed and optimized through iterative R&D cycles that evaluate how the formulation behaves under controlled conditions. We then conduct robustness testing to ensure the cycle tolerates normal process variability without risking collapse, instability, or inconsistent drying. Finally, the process is scaled and prepared for production transfer with defined parameters, documentation, and monitoring controls that support consistent and defensible manufacturing. The goal is not simply to develop a cycle that works, but one that continues to work reliably as the process scales and does not build additional risk or variation into the system. Scaling introduces additional complexity and new sources of manufacturing risk. As batch size increases, the thermal mass of the system also increases, which can change how heat is transferred through the product and how quickly ice can sublimate. Loading patterns must be carefully controlled because variations in vial placement or product depth can affect drying rates across the chamber. For example, edge vials may receive more heat than center vials, potentially pushing some product closer to collapse temperature while others remain under-dried. Shelf mapping helps confirm that heat transfer across the lyophilizer is uniform and that all positions in the chamber remain within acceptable operating limits. Without this characterization, temperature gradients between shelves or across the chamber can lead to uneven drying or inconsistent residual moisture. Operational discipline also becomes critical when moving from small R&D runs to larger production batches. Small differences in fill volume, stopper placement, or loading configuration that are insignificant in development runs can introduce variability at scale. Managing these factors through controlled loading patterns, shelf characterization, operator training, and standardized operating procedures helps reduce scale-up risk and ensures the process remains consistent as production volumes increase. Diagnostics applications place especially strict requirements on stability and reproducibility because these materials directly influence clinical test accuracy. Controls and calibrators serve as reference anchors for diagnostic systems, meaning any instability or variability can introduce measurement error across an entire testing platform. From a risk perspective, even small changes in activity, moisture content, or reconstitution behavior can lead to drift in calibration curves or inconsistent control performance. Lyophilization helps mitigate this risk by stabilizing sensitive materials, slowing degradation pathways, and maintaining consistent moisture levels across lots. By controlling these factors during development and manufacturing, the risk of performance variability in the field is significantly reduced. When Catachem talks about scaling without compromise, we mean that key attributes remain consistent. Product temperature stays within validated limits. Residual moisture stays within specification. Documentation remains complete. And post-market monitoring continues to verify product performance. Maintaining control of these factors throughout the development and scale-up helps reduce technical, manufacturing, and regulatory risk, while ensuring the process remains reproducible and defensible over time. Catachem helps partners reduce technical, manufacturing, and regulatory risk. We support formulation and cycle development. We translate processes across scales and formats. We operate under ISO 13485 quality systems with CGMP compliance targeted for July 2026. And we support documentation, traceability, and risk analysis throughout development. Lyophilization is often described simply as freezing and drying. In reality, it is one of the most precise manufacturing processes used in life sciences. When the science of freeze-drying is combined with disciplined documentation, structured risk analysis, and a robust quality system, the result is not only a stable product, but a defensible manufacturing process. Thank you for joining today's webinar. We'd be happy to take any questions.
Okay, thank you, everyone. I'm sorry about that little hiccup at the beginning. I promise we practiced this on Monday and everything worked just fine. So I'm not sure exactly what happened. If you have any questions, please put them into the Q&A and I can answer them live as we go. So first question is, how does Catachem take care of contamination risk and scale up like proteases or nucleases? So great question. We have documented SOPs for cleaning in between products. Typically, we will do a double clean with a bleach or bleach similar substance, followed by a 70% isopropyl, then a water wash for the benches, and then a repeat of the isopropyl. And that typically takes care of most proteases and nucleases. For items that are stubborn, like proteinase K, we repeat that cleaning process 2 to 3 times. It hasn't been an issue yet, but we haven't done a ton of work with some heavy proteinases or nucleases. Where applicable, we will use nuclease-free and pyrogen-free plastics and glassware, sterile wherever we can. But beyond that, we are targeting for, it says July in the presentation there, but it's probably more towards Q3, Q4 of this year for a ISO certified level 5 clean room. And in there, we haven't written the SOPs yet for all the decontamination procedures, but we should be able to deliver a much cleaner environment than we currently are when we get to that CGMP and have that clean room installed. Any other questions? Okay, another question. How do the protocols vary, and usually what's the time required? So just ask a clarifying question for that. I assume you're referring to the lyophilization recipe and the cycle. If so, then it's based on the product itself and fill size, right? So a 5 ml fill in a 10 ml vial, that's a heavy salt solution, has a much different recipe than something that is sugar-based in a 1 ml vial with a half ml fill. Obviously the smaller vials and the smaller fills There's greater surface area for the shelves to cool, and the evaporation and sublimation happens faster. Typically a 5 ml or a zero point 0.5 ml fill is anywhere between 24 and 36 hours. Whereas you get up to a 5 or a 10 ml fill, you're looking at 4, 5, even 7 days, depending on how aggressive the cycle can be. So the long and short of it is that it's a non-answer. It's really product dependent, and we work that out for every single product that comes through the door. Another question, how do you validate the process for each batch of different products? So our typical routine is we will ask the customer or whoever's bringing the product to us for the chemical constituents, how it's made, what the buffer system is, if they have any GCMS data on it, are the critical temperatures known, basically just a data gathering session about what the actual product is. From there, we can do some research and see, all right, is this an established system that's, well known in the lyophilized world? A 5% sucrose solution is very easy to work with. It's been around for decades now in the lyo world. And, we know going in right away that, all right, it's probably going to be a 3-day run, XYZ temperatures, XYZ pressures. If it's something that is not well documented, then we move immediately into R&D. We'll take a small sample and run it on our research instrument. The research instrument that we have is much smaller than our production ones. The condensing capacity is about 1.5 liters. So you can fit maybe 30 of the smallest vials and 15 to 10 of the larger vials in it. And we will develop a recipe that, all right, we think based on any of the research and information we have will work. And then we take a look at the data that the R&D instrument provides. The R&D instrument will give us an idea and an estimate of what the TG or the eutectic points are. And then based on the cake morphology and the residual moisture testing that we perform afterwards, we will optimize or refine the recipe that we ran initially and iterate that until we have found a recipe times pressures that work for this product, that particular product. And then ultimately, it will go back to the customer or downstream testing to make sure, all right, we have a dried product and does it work downstream, which is the most important part. Hopefully that answers your question. Great question. So the question is, how do you determine the process is PPQ ready? So we typically run 3 to 6 PPQ runs before we move into production. When we do iterative R&D, it takes us, anywhere between 3 to 15 runs to optimize and isolate a recipe that works for this product. And then we scale. So the nice thing about our R&D instrument is that the recipes are directly translatable to our production machines. They're all made by Millrock and they're designed that way. So we have a very high rate of success from going up to R&D scale to a production scale. Before we go into the PPQ, we will scale up into the larger instruments. Say the end result is a batch of 5,000. We'll do 1,000, 2,500, then 5,000. And then if the 5,000 works for the initial run, then we are ready for PPQ. And that's assuming that all of the QC passes, through the scale up and into those PPQ. Residual moisture is there, cake morphology looks good, reconstitution times are okay. We can measure things like pH, osmolality, turbidity, conductivity, all as potential indicators of degradation due to the lyo process. So you take those measurements pre and post lyo, and if your turbidity is 10x what it was before, then you have a reconstitution problem and the cycle is not effective. And all of that is performed in the R&D as well as pre-PPQ. And then we isolate a batch record or a SOP for the product. and then perform our PPQ runs to that SOP and make changes as necessary. And hopefully you end up with 3 to 6 PPQ runs that are all consistent with each other and pass the statistical testing, CPK, PPK, things like that. Okay. If there are no more questions, we'll give everybody a few minutes back. I apologize for the technical difficulties that we had this morning. I'm not sure what happened there. If you would like a copy of the presentation, just shoot me or David an email. They should be on the Zoom link invite as the hosts. And if you have any other questions, feel free to contact me or David, and we'll get them answered. I appreciate your time today. I appreciate you joining. You guys enjoy the rest of your day. Thank you.